everyone, I am Anu and I did my doctoral degree from Christ University, Bangalore. My study was titled Graphene and Graphene Enhanced Nanomaterials from Biological Precursors, its Synthesis, Characterization and Proliferant Applications. Graphene, as we have all heard, is a wonder material with exceptional properties, and its great deal of application is what drew me to it. But we could see that most of the time, graphene derivatives were produced from fast depleting sources like fossil-based hydrocarbons, and also involves as hard as processing routes. In order to meet its growing demand sustainably, it is essential that we find precursors and synthesis protocols that are economical and environmental friendly. When we started looking for such renewable activated carbon sources, it struck us that what better precursor there will be than charcoal. Hence, we decided to try coconut shell charcoal and wood charcoal as our first precursors. Another agricultural waste we explored as a recyclable source of graphene was sugarcane bagasse. We also selected colocasia leaves and lotus leaves that are famous for their nanoforms. All these were carbonized to form our set of starting materials. In order to synthesize graphene from these materials, we went for the age-old trusted Hummers method, but modified it to avoid the use of harmful reagents such as hydrogen hydride, sodium borohydride, etc. And to much of our pleasure, the reduced graphene oxide we obtained was as good as that procured from the standard reference carbon black that we used in our study. All said and done, what is science if we do not give back to the society? One of the critical areas where graphene has immense potential is as antibacterial agents. With the rising threat of mutated antibacterial resistance, something needed to be done. Therefore, we embarked upon this multidisciplinary journey towards creating an efficient bactericidal agent with multiple destruction pathways. As we combed the literature, we observed that incorporating metal oxides on graphene skeleton greatly enhanced the antibacterial activity through synergistic effects. We were in search of a graphene-based antibacterial agent that can perform well in natural lighting so that photocatalytic deactivation due to active site blockage would not be an issue for us. It was then we came across reports on the antibacterial activity of tin oxide under dark conditions. So we went ahead with incorporating tin oxide into graphene frame in the hope of producing effective bactericidal composites, and it paid well. So we produced 3 variations from each of these 5 samples and reference carbon black, that is carbonized precursors, graphene oxide after Hummers treatment, and the composite. The formation of these derivatives were confirmed with various characterization methods. To check their antibacterial efficacy, instead of going for the regularly used gram-positive or E. coli strains, we took a highly potent gram-negative escape pathogen, Pseudomonas aeruginosa, as the model bacterium. We used antibacterial drugs like clindamycin and a more potent cephalexin as controls. Our study ascertained that the nanocomposites had higher antibacterial activity compared to its oxidized and precursor forms. All the synthesized materials showcased bactericidal effects higher than clarithromycin, and among them, composite from colocasia leaves showcased an activity equivalent to that of the antibiotic cephalexin. The varied efficacy between samples synthesized under identical conditions points to their precursor dependence. Also, antibacterial effects of our samples were either equivalent or superior to that of graphene silver nanosystems. We found that these higher antibacterial effects were stemmed from various factors such as particle size, morphology, and synergistic effects of these materials. The fact that all our samples were showing excellent antibacterial efficacy under natural illumination increases their commercial applicability. Our research has paved the way for use of nanocomposites obtained from green precursors via green routes as economic and ecological safe alternatives for the traditional harsh disinfectants. As a first step towards wide visibility of the invention, we filed and published a patent for part of our work, after which the same findings were featured in the journal Chemistry. The results of charcoal samples were published in scientific reports, where it was featured in the scientific reports top 100 articles of 2019. Other studies were published in journals such as New Journal of Chemistry and Materials, Chemistry and Physics. So before bringing it to the market, we plan to complete the human clinical trials of our material. Since the synthesized nanostructures were showcasing tunable photoluminescence, feasibility in using them as bioimaging probes is a promising research area of the future. Thank you.